Hello everyone, welcome back. I hope you all are doing great. Today we'll start with our plant pathology lecture series. This is the first lecture of our new lecture series. So, this picture gives us a gross idea about how pathogens enter into susceptible host and thereafter penetrate into the tissues and colonize there and thus establish disease. You can see invasion of susceptible host occurs through natural openings or some other type of opening. Then infection of leaf and vascular tissues occurred. Then development of V-shaped lesion on the leaf. Then the pathogen progresses into the stem and root, and finally causes systemic infection of plant. Then the seeds become infected, and this is how the uh, cycle perpetuates. Now we will discuss this in detail. Okay. So first is our recognition and entry of pathogens into the host cell. See, host pathogen battle is something very interesting. Here, one will win, the other will lose, and sometimes there is some mutualism also observed here. But this infection or microbial infections in plants remain a major challenge in agriculture. This has to be addressed with utmost care. So, coming to the point, interactions between plants and pathogens commence with some kind of communication between them. If during this contact some specific requirements are met, the so-called recognition between both partners can take place. There has to be certain type of requirement that has to be met. Otherwise, there is no recognition between both the partners. This cannot happen until and unless some requirements, some specific requirements are met. Recognition implies that the onset of a series of biochemical reactions is triggered in one or both partners. The recognition event consists of the specific interaction of a particular signal with its sensor or receptor, the latter being located on the surface of one of the interacting partners. This is called a signal-sensor reaction. The signal can be either chemical or physical, and signal recognition requires very close contact between plant and pathogens. This cannot happen, recognition cannot happen if they are distantly located. So, close contact is one of the first and foremost criterion that has to be met in order to have successful recognition, entry, and colonization. So the infection process has got three parts to it. Pre-entry, entry, and colonization. So before entering into the host, there is a phase called the pre-entry phase. Before a pathogen can penetrate a host tissue, a spore must germinate and grow on the surface of the plant. In the case of motile pathogens, they must find the host and negotiate its surface before entering the host. Well, some pathogens, they develop specialized penetration structures like Aprosoria, while others utilize the pre-existing openings in the plant surface such as wounds or stomatal pores. This pre-entry event is followed by entry phase, and finally, if these are successful, then the pathogen is able to colonize into the susceptible host. So we'll first talk about adhesion. The pathogen needs to adhere to the surface of the host, isn't it? Until and unless they adhere, how will they enter? So we'll talk about adhesion process first. So this is a part of pre-entry itself. So adhesion means what? To remain adhered to the host tissue. In fungal substratum adhesion that occurs on plant host surface before penetration, this event that occurs, no, this event serves multiple functions, such as like it keeps, if we take one by one, it keeps the propagules from being displaced by not being blown or rinsed by water or ringed from a potentially suitable environment. So adhesion should be firm enough. The propagule should not be blown or washed away. Next, it requires it is required for host penetration via mechanical pressure, thigmodifferentiation, thigmotropism. Okay. It facilitates interaction between pathogen and host and increase the surface area of contact with its host. So surface area of contact is very important. If it becomes too less, then it is difficult for the pathogen to interact with the host. So increasing the surface area of contact is very important. Next, limits germination to potential host tissue, which is required for contact stimulated germination. The initial contact between the infective propagule of parasites and host is called infection. Please do not use these type of uh, writings in your exams. Several fungi use topographical cues on the plant surface to guide them towards a likely stomatal site. Once the hypha reaches a stoma, volatile compounds escaping from the pore appear to provide a signal for the formation of specialized penetration structure called aprisorium. See here, this is the spore. The spore starts to germinate once it feels that the environment is friendly. After germination, it forms a structure called aprisorium. It forms a structure, it's a specialized penetration structure. Aprisorium is a specialized penetration structure. Sugars, amino acids, and minerals secreted by plants at the leaf surface non-specifically trigger spore germination or provide nutrition. This is important. The spore needs nutrition to germinate. So provide nutrition for the pathogen. Sometimes spore do not germinate in the absence of these substances. So these substances are of premier importance. Now, pathogen development is influenced by temperature, moisture, light, pH, aeration, nutrition, all these factors. Next, coming to entry. Pathogens exploit all available possible way to enter their host. Although individual species of pathogen tend to have a preferred method, like fungal pathogens often use direct penetration of plant surface to enter the host, which requires addition of the plant surface, which we have just discussed, followed by application of pressure and then enzymatic degradation of cuticle and cell wall in order to overcome the physical barriers presented by the host plant surface. So let's come to penetration. What happens here? Pathogens will exploit every possible way to enter their host. The stimuli provided by the host for germination, growth, and differentiation of infection structures can be hydrophobicity, hardness, chemical components, and topographical features of the host plant. So, these are important: hydrophobicity, hardness, chemical components, and topographical features. These from the host end, these are important. The wax on the surface of the aerial parts is a rich source of diverse compounds that help in propagule, germination, and differentiation of infection structures. The topologies of the plant surface provide signals to many fungal pathogens, like the rust fungi enter through the stomata, their topology triggering the development of infection structures. Germ tubes of Puccinia graminis orient themselves at an angle of 90 degrees to leaf veins, which, owing to the manner of their distribution, maximize the chance of the tube encountering a stoma. So these are certain means through which they enter. Now, apart from this, the aprosorium, as we have discussed, plays an important role. The cell wall of the aprosorium often becomes impregnated with melanin, making it watertight and capable of containing high turgor pressure that builds up within the aprosorium. The point of the aprosorium that is in contact with the cuticle is called the penetration pore. So if we write that, the point of the aprosorium that is in contact with the cuticle is called the penetration pore. And the wall is the thinnest at this point. The increasing turgor pressure causes the pore. The increasing turgor pressure often, the result in formation of a penetration peg which applies huge pressure to the host cuticle and cell wall. This is basically then for pressure application on cell wall and cuticle. Right. Next comes the event of post penetration. So what's happening after it penetrates? Invasion and colonization. See, infection is the process by which pathogens establish contact with the susceptible cells or tissues of the host and derive nutrients from them. Right? A parasitic relationship is formed between host cytoplasm and parasite cytoplasm. And during infection, pathogens grow and multiply within the plant tissue. Invasion of plant tissues by the pathogen and growth and reproduction of the pathogen, which is called colonization, are the two concurrent stages of disease development. Fungi spread into all parts of the host organs, either by growing directly through the cells as an intracellular mycelium or by growing between the cells as an intercellular mycelium. Intercellular mycelium. Now, during establishment, pathogen produces different substances which include enzymes, toxins, growth hormones, and polysaccharides, which will help in colonization. See, this remember, a successful infection requires the establishment of a parasitic relationship between the pathogen and the host. Once the host has gained entry into the plant. We will discuss this topic in our upcoming video, but just letting you know, there are two broad categories of pathogens which we call as biotrophs and necrotrophs. Biotrophs are those that establish an infection in living tissues, while necrotrophs are those that kill the cells before colonizing them by secreting toxins that diffuse ahead of the advancing pathogen. Now, these two kinds of pathogens are also sometimes known as snakes and thugs because of the tactics they use to acquire nutrients from their host. So this is how the process of colonization occurs. So this is how we come to the end of our lecture today. Hope you have enjoyed learning something new. If you have any confusion, any doubt, please feel free to get back to me. Till then, bye-bye.